Okay. The Delta-proteobacteria are a little bit more homogeneous. The other groups we talked about, there's phenotypes all over the place. The Delta-proteobacteria fall into three phenotypic types. There are the sulfur reducers. These are organisms that grow by respiration, but not aerobic respiration, by anaerobic respiration, using sulfate, usually, as the terminal electron acceptor and generating sulfide as a result. And so these things are taking organic material, oxidizing it to CO2, and dumping the electrons on sulfate to make elemental sulfur, or usually hydrogen sulfide. These are the organisms that produce the black layer at the very bottom of your Winogradsky column, because they're hydrolyzing cellulose, or using sugars released from that cellulose by other organisms, and generating CO2 and sulfide, which binds to iron in the soil to create iron sulfide, which is black. Also in this group are the myxococci. These things are a form of slime mold, and so they're bacterial predators. They eat other bacteria and form multicellular sporing bodies. These things are aerobic heterotrophs, common in soil. And then lastly, there are the internal bacterial predators, Bacteriovorax and Dellavibrio, which infect other bacteria, living typically in the paraplasm, and they're parasites of other bacteria. So here's desulfovibrio desulfuricans, a traditional, quite common sulfur reducer, and they have this kind of banana-shaped cell that you commonly see in these environments. These things are most usually found in sediments where you have organic material like leaves and things, complex polysaccharides settling to the bottom in the anaerobic zone, and they're using sulfate from seawater or that sort of thing to generate sulfide. And so this is why estuaries, for example, have black sediment. When you go to a salt marsh or an estuary, you see that black stuff, or a beach for that matter, and dig down into the soil. If there's organic material in there, you'll see that it's black. Here's Myxococcus. This is a member of the aerobic fliers. These things live as individual cells in the environment and feed on other bacteria. So they secrete hydrolases and lysozymes, lyse surrounding bacteria, and suck up the organic material that's released from that. Although they're unicellular, they usually act in groups. And so a collection of these organisms will swarm, they communicate and move together, and swarm over other bacteria, eating them as they go. There's some great videos of this. When the food runs out, though, they come together. Millions of them come together. They swarm, communicate each other, come together, and form a fruiting body. And so the cells pile up, the cells differentiate, push up into the air, and they undergo a terminal differentiation. So the cells at the top in this fruiting body form what amount to spores. Not endospores, they're not heat resistant or anything like that, but a quiet survival stage of the bacteria. Things that can be disseminated in the water or the air. The stalk, the cells that hold this up into the air, are terminally differentiated. They'll never divide again. They're incapable of proceeding. And so this is in realistic terms, this is a multicellular organism at this stage, because you have distinction between germline and vegetative cells. Myxococcus forms a very simple fruiting body. There are other species that form highly complex, branched, really cool-looking fruiting bodies. This is Dellavibrio bacteriovorans, and this is E. coli. This is a host for this organism. These things are quite small. They're in free-living form, they're swarmers. They're metabolically inactive. They just swim around, and when they touch a bacterium, and they're looking for N-acetylglutamic acid. I think that's the amino acid they're looking for. Then they burrow into the cell. They pierce the outer membrane and push themselves into the periplasmic space. And they grow, sucking nutrients out of the host cell. They don't actually divide. They get longer and longer. They form kind of a helical thing in the periplasm until ultimately the entire host is destroyed and used up. This filament inside the periplasm then undergoes a synchronous division, divides into usually 8 cells all at once, and those are released into the environment as swarmers. And this process repeats itself all over again. Normally these cells are too small to see with a microscope, but occasionally you can see these tiny, tiny cells swimming around in a culture. Usually we see it in Bacillus cereus. Bacillus cereus is one gram-positive organism that is susceptible to infection by this thing. And so if your Bacillus cereus culture isn't growing very well, and you see these little tiny things swimming around in a wet mount, you may have Delta-vibrio.